Hi, everyone. My name is Jimmy Liao. I'm an associate professor of biology. And on behalf of myself, Director Mark Martindale, and the rest of the Whitney faculty, welcome to the evening at the Whitney. This is a seminar series that we have once a month to bring in scientists from UF and beyond to talk about timely science and their research. The Whitney Lab, if some of you may not know, was established in 1974 as a full-time research institute of the University of Florida. I know this because that was the year I was born. So it's as old as me, or I'm as old as it. And originally it was for molecular biology and neuroscience. And since then, it's really expanded to include regeneration, biomechanics, development, conservation of the environment, sea turtles, aquaculture. So it really has expanded quite a bit. And there are 11 full-time faculty at the Whitney Lab, and we all play an active process in mentoring students from the K through 12 outreach level, all the way to undergraduates, to graduates and postdoctoral fellows. So tonight, it's my distinct pleasure to introduce Dr. Jim Strother. Jim received a BA in physics in Berkeley, at Berkeley. He then completed his PhD in Matt McHenry's lab at UC Irvine, a close colleague of mine, and he studied respiratory physiology of fishes. He then got really interested in animal behavior, and he went to Howard Hughes Medical Institute, the Genelia campus, and he looked at neural computation of vision in flies. He then started his own lab at Oregon State University. And after a few years there, we were lucky enough to hire him at the Whitney Lab. The timing was not great because he had to set up his lab during the COVID pandemic, but he's made impressive progress. He is spearheading the Whitney's new Center for Biological Imaging. He also received a very prestigious Paul G. Allen Foundation grant, along with other Whitney faculty members. And this is the first time this prestigious grant has been given to any UF faculty team in the history of UF. Yeah, yeah, please applaud. It's the kind of grant that you can actually put on your email signature. You can say Paul Allen Fellow. So that was great. So there's a saying that you never want to be in the room where you're the smartest person. This is not going to be an issue if you're talking with Jim, because Jim is one of the most classically trained young biologists that I know of. He's equally adept at chemistry, deriving mathematical equations, building electronic circuits, building microscopes, recording from neurons. And he leverages this prodigious intellect to study questions at the interface of complexity and animal behavior. So one theme in his research is clear thought as well as clear organisms. He's going to talk to us and take us on a journey inwards into the brain today. And so I suggest you buckle up. Jim.
Thank you. All right, great. So thank you for that very generous introduction. I'm really excited to have the opportunity to speak with you guys and tell you a little bit about the research that we've been doing. I'm going to try and put it into a broad context so that you can see not just some of the research that we're doing, but also kind of the state of the field and where the field in general is headed. Because I think that there's a lot of sort of exciting opportunities within the fields of neuroscience at the moment. So with that in mind, what I want to do is start by taking us back to the beginnings of neuroscience and talk about some of like the early advances that paved the way for the work that we and others are doing. And so I want to start with some of the work that was done by Luigi Galvani back in the 1700s. He did some of the early work in neuroscience, making the connections between physiology and cognition, how the brain works. And his advance was to recognize that if you took this is one of the original plates from a manuscript, was that if you take the severed leg of a frog and you connect it to a Leyden jar, which is a type of battery, that you can get twitching of the muscle of the leg. And what that did was it established a connection between electricity, which was sort of known in the context of batteries and atmospheric electricity, lightning, and that sort of thing, with how muscles and how the nervous system worked. And then if we fast forward another couple of hundred years, one of the next major advances was done by Ramon y Cajal and his colleague Camillo Golgi. And what they did was they looked deeply into the anatomy of the nervous system. And they developed a technique where you could label individual neurons and really see all of the intricacies of the anatomy. And what that enabled them to do was to look into the brain and recognize that it wasn't a sort of homogeneous soup of neurons. And instead, it was comprised of this huge diversity of different neuron types that were all intricately connected in complex, but often characteristic ways. And this is one of the original drawings that they came up with, again, showing all of these neurons, all of the little fine processes, these little dendritic spines, and showing all of these sort of intricate connections between them. And fast forward another few years, Julius Bernstein made one of the next major advances, and that was to give us a deeper understanding for what this connection was between electricity and neurons and muscles. And what he proposed was that these changes in the electrical potential of neurons and muscles could be attributed to changes in the permeability of the membrane to different ions. So that as the membrane changed its permeability, different ions would move through that membrane and change the electrical potential across the membrane. And that these differences in electrical potential would then propagate along the neurons and then through this sort of large network that had been described by Cajal and others. This is a drawing of the device that they used. I have no idea how that's put together, but it's very neat looking. And then one of the next sort of major advance in the field was performed by Erwin Neher and Bert Sackmann in the 1970s. And what they found was that you could dig even deeper by using a technique called patch clamp. And in patch clamp, what you do is you use a pipette in order to that's sealed against the cell membrane. And what that enables you to do is to record not just changes in the electrical potential at the cellular level, but even finer at the molecular level. And what that allowed people to do was to understand how these changes in membrane potential connected to the actual molecules, the actual ion channels that were mediating these effects. And with all of these tools kind of in the toolkit, there was this sort of flurry of activity in the next couple of decades where people were able to use these methods in order to elucidate how different types of neural circuits were put together. And the most sort of impressive of these results occurred at the periphery of the nervous system in the sensory structures. And so this is just a sort of example of how the vertebrate retina is put together. And there's a vast amount of information known about how the photoreceptors connect to bipolar cells, how those interactions are mediated by amacrine and horizontal cells, and how that signal is propagated to the retinal ganglion cells. Just tremendous, we have a tremendously deep understanding of how these neural circuits are put together. And there's a similar level of understanding of many different sensory systems and other elements of the peripheral nervous system. But one area that's proved much more elusive and much difficult to tackle is the brain, is the central nervous system. Certainly we have an understanding of what the different brain regions are functioning for. We have a general understanding of what different types of neurons are in which regions, but we certainly don't have the same sort of cellular level resolution, the same sort of cellular level understanding of how the brain works. We certainly don't understand how complex the neural circuits that mediate complex behaviors and how the sort of emergent properties of these incredibly complex diverse circuits give rise to cognition and other types of sort of more higher order behaviors. And so why is that? Well, I mean, it's a fundamentally simple problem, which is that most of the techniques that have been used historically within neuroscience focus on individual neurons and recording from one neuron at a time. So if you're looking at a relatively simple circuit, by analogy, this is a simple electrical circuit. This is sort of roughly similar to what you might see at the in a sensory neural circuit. A small number of components connected in a relatively simple way, and you can sort of march your way through the circuit, recording from each of those components. And by doing that, it's not easy, but if, you know, with some effort, you can piece. Did I just lose the mic? There? Still? All right. Okay. You can piece together how that circuit is working. But when we move into the central nervous system, into the brain, we get circuits that look more like that. And you can imagine that in theory, maybe you could make progress recording from one component at a time, but just you get this order of magnitude, orders of magnitude increase in computational complexity, in number of neuron types, in the connectivity of the different neurons, and recording from individual neurons one at a time no longer produces the same type of progress that you can get looking at the, as you would looking at the simpler neural circuits that are available at the periphery of the nervous system. And so what I want to talk today about is some of the approaches that we use for trying to get at those types of circuits, trying to move from the periphery of the nervous system and trying to understand at a cellular level what's going on within the brain, within the central nervous system. And the approach that we've taken and others have taken is to look at this impossible problem and to ask ourselves, what's the simplest version of the impossible problem? And one of the ways of looking at that is to just think about how big different types of brains are. And you can sort of quantify that in terms of number of neurons. It's a sort of rough measurement, but it's a measurement. And so if you look at a human brain, there's, on the order of 90 billion neurons. Dogs might be 2 billion, mouse 70 million, adult zebrafish 10 million, larval zebrafish 100,000. So that's about the smallest vertebrate brain that you're going to come up with, is 100,000 cell types. And that's about 1 million times smaller than what you would see in an adult human brain. So that seems great. 1 million times smaller seems like a more achievable problem. If you look at invertebrate organisms, you'll see smaller brains. But when you're thinking about if you restrict yourselves to vertebrates, then a larval fish is a really small brain. Now you might be wondering, okay, it's small, but does it carry? Does it generalize? And to add to that, we have to look at kind of what the organisms look like in what the organisms themselves look like. And so these are the kind of model organisms that my lab has been looking at. The first is an adult zebrafish, which you can see here. It's a fish that's native to India and to the streams of India. Feeds on zooplankton principally. And the other organism that we look at is larval zebrafish, which is shown here. I'm going to zoom in so you can actually see it. It's about the same size as a piece of rice. And the brain is about the same size as a fine grain of sand. But despite that small size, it exhibits a lot of sort of complex behaviors. About 5 days after the embryo is fertilized, this animal has developed to the point that it's freely swimming. It navigates its environment, it finds its own food, and it avoids predators. I've got a 5-month-old kid at home, and it can do none of those things. And so even though it's a very small organism, it's still capable of navigating its environment. It's entirely self-sufficient. It's an entirely self-sufficient organism at 5 days. And so how does the brain look? How does the brain compare to what you might see in other organisms? Well, surprisingly, the vertebrate brain is relatively strongly conserved. And so there certainly are differences, but if you look at the gross anatomy of the brain, it's fairly similar between mammals and other vertebrates. And so this is a sort of mammalian brain showing a side-on view, similar to what you would see here in a mouse. Each of these drawings is color-coded for different sort of gross anatomical regions: the olfactory bulb, pallium, optic tectum, cerebellum, medulla, pituitary, hypothalamus. And what you'll see is that there certainly are differences, but the sort of gross anatomy of the brain is relatively similar between vertebrates, and is really quite similar between mammals and bony fishes and amphibians in particular. And that's true even if you zoom in a little bit. Even if you look at finer anatomical structures, we see very strong similarity between the different regions. We see similar neuron types, and we see them wired together in certain ways, in similar ways. Now, obviously, there are differences. Fish aren't people, people aren't fish. But there are strong similarities suggesting that the basic principles of how the brain works are conserved over a fairly large diversity. And in particular, when we look at the brain of a larval fish, we see that it has similar anatomy. So this is the brain of a larval zebrafish. The snout would be sort of over here. The back of the head would be somewhere over here. And then the right side of the animal would be on the top, and then left would be on the bottom. And what you see is that it has all of the same sort of gross anatomical structures that you would see in adult fish and adult mammal. Olfactory bulb, telencephalon, diencephalon, optic tectum, cerebellum, medulla. And again, if you look into each of these regions, you see similar neuron types wired together in a similar way. In many ways, it really is a sort of canonical version of the vertebrate brain, just miniaturized. The smallest sort of canonical vertebrate brain that you could come up with. And so this is the model that we've been trying to use in order to understand how the brain works. And the specific questions that we've been focused on in my lab is trying to understand how emotional state modifies the behavioral responses of animals. In animals, we would typically call it motivational state or behavioral state, but fundamentally it's referring to the same thing. The idea that in different states, the animal will perceive and respond to stimuli in different ways. And one of the specific types of behaviors that we've been looking at is a response to stressors. And so I wanted to give you an example of how different states alter that response to a stimulus. And so I want you to ask yourself, you're walking along the road and it's Florida. And so a cloud opens up and it just starts dumping rain. What is your response? Run, right? You run. You stick something over your head and you run. Now, what if you find yourself walking down a road, there's nothing else around, it starts raining, you look around, there's nothing to run to, you've got nothing in your hands, you've got nothing to cover your head with. What's the next response? Yeah, you're just gonna get wet. You accept the fact that you're gonna get wet. And all of a sudden this rain that was this really strongly aversive stimulus that you had to get away from becomes this thing that you're fine with. You're just gonna deal with the fact that you're gonna get wet. This seems like a sort of uniquely human response, but it's actually reflective of behavioral responses that are nearly universal. Within the sort of behavioral sciences, we would say that the acute response to aversive stimulus typically causes what we call an active coping or avoidance behavior, which is that you get this negative stimulus and you just try and get away from it. But the response to what we would call an inescapable aversive stimulus results in what we call passive coping or sedentary behavior. You hunker down and you just try and ride it out. So one of the first questions that we asked was, do these larval zebrafish with their little tiny sand grain brains demonstrate these same types of behaviors? And so the way we went about asking that was to put the animals into a small dish. We record their movements. We use software to automatically track the animals. And we presented them with a mild aversive stimulus. And the stimulus that we use is mustard oil. the same compound that you would find in mustard or wasabi. We give them at a very low concentration, so it's barely perceptible, but fish don't really like mustard, and so it's a sort of mildly aversive stimulus to them. And what we find is that typically they swim at a sort of routinely at a specific rate, but when we administer this stimulus, we see this jump in swimming speed. And this is entirely reflective of what we would consider active coping or avoidance behavior. The animals sort of swim around the dish looking to find water which doesn't contain that stimulus. But interesting, what we see is about after 4 to 5 minutes, we see the swimming rate decrease back down, not to baseline levels, but actually below baseline values. And then it persists at this sort of below baseline value for about an hour or so before the animal recovers back. And this phase is entirely consistent with what you would expect as for passive coping in response to this. So these animals, even with their little tiny brains, demonstrate the same types of responses to stressors that animals up to and including ourselves do. All right, so here's the next challenge for you. So you're not feeling great. It's the weekend, you're trying to figure out what you want to do, but you're not really feeling good. You're feeling sick, maybe you're recovering from a cold, you're feeling tired, you're just feeling like burnt out and not great. Do you decide you want to go for a hike in a Ocala National Forest, or do you hang out at home and watch a movie? Who wants to go for a hike? Okay, who wants to hang out at home? Yeah, everybody wants to hang out at home. And again, this is reflective of very general types of animal behaviors. We would say, as a general rule, animals that are unstressed and unfatigued display more exploratory behaviors. They'll seek out novel environments. Which is often looking for different types of resources. They spend more times in well-lit environments. Those environments certainly offer more opportunities for finding food, potentially finding mates, although also there's a greater predation risk. And so in animals that are displaying more anxiety-like or fatigue states, typically demonstrate what we would call a refuge seeking, which is avoiding novel environments, seeking out familiar, often darker environments, which are contained. And if you think about it in a sort of evolutionary context, might have less predation risk. And so we asked, do our animals display this type of behavior? So to look at this, we put them into a chamber that had a light side and a dark side. We allowed them to kind of freely maneuver through it. And then we measured the exploration index, which was a measure of how much time they spend in each side. So if the animals are exploring each side equally, the exploration index is 1. If they're spending all of their time on one side, then that exploration index is 0. And what we find is that the animals start off, they're in a pretty good way. They'll explore their entire environment, spend about as much time on both sides. But when we introduce that mild stressor, the animals will no longer explore their environment. They'll sort of pick a side and hunker down and wait for that aversive stimulus to go away. And so just like you might, these animals sort of display a transition from exploratory to refuge-seeking behavior. And so the next question that we asked is like, okay, that's great. These animals really do seem to display the same types of behaviors that other organisms do. Can we dig into the brain and look for how these responses are being mediated by different neurons and different neural circuits? And one of the challenges is that they have very, very small brains, but 100,000 cells is still 100,000 cells. And if you're recording from them one at a time, it's still a challenge. And so what we need is a better way of recording large populations of neurons. And the approach that we use is optical neural activity recordings. And so those largely rely on fluorescent indicators. And so fluorescent indicators are chemicals that absorb at a shorter wavelength of light and emit at a longer wavelength of light, so often absorbing in the blue and then emitting in the green. And I want to do a little demo to show you why that's useful for recording from different neuron populations. So Turn off the lights here. And that, okay. Let's see how this works. Right. So what I have here is a flashlight, science flashlight, which is at a blue wavelength. And what I want you to see is obviously it's a blue flashlight. If you point it at something, then it turns blue. But what I have down here is a vial of fluorescent fluid. And what you see is that it's absorbing the blue light and then re-emitting that as a green light. And so if you're imaging from a biological sample that contains a lot of sort of complex optical structure, what that allows you to do is to, if you can label your compound with that fluorophore, then even though there might be a lot of complexity and structure in the room, everything else in the room is going to be blue. If you shine a blue light at it, you're going to get blue light back. But if your neuron is labeled with a fluorophore, then you shine blue light at it and you get green light back. And so even in large populations with complex structure, you can still localize individual cells. And then the other thing I want to point out that'll become important later is that it's wavelength dependent. And so if I use a red light source, again, red light in, red light out, but my fluorophore is no longer active. I put red light in, I get red light out. And I'll explain why that's important later on. All right. And so some very clever colleagues in other labs have developed not just fluorescent indicators, but indicators that indicate a level of neural activity of the cells that contain them. Typically, these indicators work by being calcium dependent. And so they'll bind to calcium. When a neuron is active, it tends to have higher levels of intracellular calcium. The fluorophore will bind to this calcium and then kind of shift into an active state. And so you'll go from something that something that isn't glowing to something that does glow. And so you can use that fluorescence as a readout of the activity of a neuron. To give you an idea of what that looks like, here's a quick video in a loop of a collection of cells on a plate. And so you can see that you can use these techniques to go from recording one neuron at a time to recording hundreds of neurons at a time. Large populations of neurons where you can start to look at and start to understand some of the diversity of the responses. But one of the challenges that remains is that these techniques, the sort of traditional version of that technique, doesn't work especially well when looking at three-dimensional structures. And the reason for that is that the light coming in is going to get focused into a cone, but it's going to excite all of the material above that imaging plane and all of the material below. And so in this direction, there's very little resolution as to what you're looking at. So in TD, two-dimensional space, you're great. But if you try and look at three-dimensional brains, you have this sort of substantial loss of resolution in the axial direction. And one of the kind of major advances in resolving this came in the 1930s by a theoretical physicist by the name of Marie Goeppert-Meyer. And what she recognized was that if you excite the sample at these longer red wavelengths, what you get is no fluorescence. Just like what we saw, if you use a red wavelength, you get no fluorescence. But what she realized was that if you hit the sample with an extremely intense beam of light, then as the light is traveling through the sample, it induces a sort of transient change in the mechanics of the molecule that allow it to accept 2 photons of light instead of the typical 1. And so that allows it to absorb that light and produce fluorescence. Now, about 60 years later or so, this sort of insight into the theoretical physics resulted in a technique which we know as a multi-photon microscopy, where a laser beam is directed into the sample and produces excitation within the image plane without producing any of those tails in the space above or in the space below, allowing a really high resolution, deep penetration imaging of complex biological structures. And what's interesting about this is how reflective it is of the sort of scientific process as a whole. That you have this sort of scientific advance in the 1930s, and then decades go, decade by decade roll on, little advances happening in other fields until however many years later, all of those advances come together and they produce a new application space that none of the original investigators were able to foresee. So that really speaks to why it's important to do basic research, because it establishes a foundation that other applications rely on. All right, so if we take all of these pieces, if we take the small brains, if we take the fluorescent indicators, if we take the multi-photon microscopy, then what we can do is put those together in order to do some really exciting science. What we can do is, instead of recording from one neuron at a time, we can record from every neuron. We can record from every single neuron within the brain of the animal. And we can do it with cellular resolution. So we're not recording from this brain region, does this thing at this time. We're recording with cellular resolution the activity of every single neuron in the brain. And the specific workflow is that we use fluorescent indicators that are loaded into the embryos. We use a zebrafish larvae. Their head is embedded in a block of gel, so they don't really move around too much, but they're small animals, so they're perfectly happy to swim around, even though they don't know they're not moving. We then image the brain of the animal using multi-photon microscopy. This produces large data sets, which we then analyze using artificial intelligence and machine learning algorithms. And then we get neural activity maps that tell us about how they're processing their environment. I want to show you what some of those look like. And so this is a map showing at a little bit faster than real-time speed, different populations of neurons color-coded of an animal that is just really at rest. So no stimulus, this is just sort of like baseline rhythmic activity in the brain. And I think what you'll see is that there's a lot of activity. The brain's a busy place. And so there's a diversity of neurons sort of interacting in a diversity of different ways. You see a lot of rhythmic activity within the cerebellum. This is likely attributable to swimming types of behaviors. There's also activity in other parts of the brain that would be more associated with executive function and motivational state. But in general, there's a lot of complexity, but the advantage of this technique is that we can capture that diversity and the complexity of those responses. For the first time, we can really see in great detail what's going on in the brain as it interacts with its environment. And we can use this question to then go back to those types of behavioral experiments and look at what was happening in the brain of the animal when it was responding to that mustard oil. What happened here, what happened here, what happened here. And so we can reproduce that exact same experiment, but in a context where we can record from the brain throughout the experiment. And so I'm going to show you the raw data for that. So this is a baseline activity in the brain as the animal is, this is pre-mustard oil. It's just hanging out. The mustard oil is added. So I hope you can see there's a flurry of activity, sort of changes in specific activity of different regions. And over time, what we see is a sort of decrease in the overall level of activity and changes in the specific activation of specific neuron populations. And so what we see is very similar to what we saw behaviorally, only played out in the context of the whole brain. We see that the initial response is this sort of flurry of activity on the same timescales as what we saw for the avoidance behavior. And that's followed by a period of sort of suppression of neural activity. But it's brain's a big place still. Even with 100,000 neurons, the brain's a big place. And so to unpack exactly how this neural activity might be associated with some of the behavioral responses, we use machine learning to look for specific neuron populations that have the most predictive value, with the interpretation being that these are sort of causally related to some of the changes in behavior that we see. And when we do that, we get this type of a map. So the blue is background. Let me see if I can. Is that better? Okay. Now I want to go back and show you the other movie. Okay. The blue is showing background, and then we've labeled the red pixels or voxels as voxels that have the most sort of predictive value. And I hope what catches your eye is this population of neurons right back here. So each one of the little dots there is an individual cell. They're sort of blurring together because of the animation, but each one is an individual cell. And that's a population of neurons, which is known as the area postrema. It's a population of noradrenergic and dopaminergic neurons that has broad projections throughout the brain. And as I said, there's a lot of homology and conservation across the vertebrate brain. And so the area postrema is a region of our brain as well. And in our brain, it's also been associated with the sensing of noxious chemicals. So again, that's consistent with the idea that similar anatomical regions have similar functions. And so we can dig into that a little bit more. Because we have cellular resolution recordings of what's going on, we can look at the response of every cell within that region to the stimulus. And what we find is that there's functional diversity. So it's not the case that exposed to the stimulus, all of the neurons do exactly the same thing. Instead, what we see is that some of the neurons show a very rapid transient response, whereas other neurons show a prolonged response. I've sort of given you a histogram of the change in, or the half-life, the distribution of half-lives up here. So time is on the x-axis here, y-axis is activity. And so this transition, or this difference in the timescales between the transient and the prolonged response coincides exactly with the type of, with the timescales that we observe behaviorally for the transition between active and passive coping. And then the next step is that we can go back to behavioral experiments and look at what happens if we block the connections of these neurons to some of their downstream targets. And so we used a pharmacological manipulation to block that connection to the downstream targets. And what we find is that that reduces the overall, or the sort of baseline swimming activity, and it also blunts that response to the stimulus. So it decreases the active coping response without altering the passive coping response. Again, suggested that these particular receptors are contributing to that particular behavioral response. And so it's a big circuit with lots of complexity in our data, and the data from other labs suggests that there are other neurons that contribute to these responses in the habenula and locus coeruleus and a number of other neuroanatomical components. But I hope that you can see that using these techniques, we can really dig into these neural circuits, and we can get a handhold on what are the different neural populations that are mediating these effects, and kind of march our way through the neural circuit in order to understand at a very detailed level how they're put together. And so the next question that we asked ourselves was, so far we've been using drugs to understand the behavior and to understand how the neural circuits are put together. But we asked ourselves, can we go the other direction? Can we use what we know about the behavior and what we know about the brain in order to find new drugs? And in particular, we started thinking about applications for chronic pain and itch. So chronic pain is any pain that lasts longer than 3 months. It's really a sort of broad suite of diseases, including, is caused by untreated injury, surgery, neuropathic pain, arthritis, depression, anxiety. There's lots of causes of chronic pain. And the sort of current drug of choice for treating it remains opioids, which has many, many side effects, because it acts both at the periphery of the nervous system as well as within the central nervous system. So it has a strong propensity for inducing addiction. And so what we did was we partnered with another lab at the Whitney lab, the Luskin lab, and we decided to look for compounds that might be mediating the response of the larval fish to aversive stimuli. And so we took the workflow that we used was to look at their extract library, to take chemicals that they'd isolated from marine microbes and fungi, and then expose our zebrafish to those compounds and look at whether it changed the response to, again, these sort of mildly aversive stimuli. And if we found that they did, they would, the Luskin lab uses NMR and LCMS to try and understand the structure of the particular compound. And we then dig into the mechanism of action using whole brain imaging to identify the neuron populations that are affected by these compounds, and then in vitro cell-based assays to understand the specific receptors that are being targeted. So to give you an idea of what that data looks like, here's a response to a control. So it's a simple behavioral assay. So time is on the x-axis. Different individuals are kind of shown as different rows. And black pixels are going to represent no movement, and then brighter pixels are going to represent higher levels of movement. And so we initially expose the animals to, in this case, a control, and we see there's no movement. The animals just sort of hang out at the bottom of the dish. If we expose them to the mustard oil, then we see, similar to what we saw before, is a sort of increase in movement and then a decrease back down to a baseline. And we can use that to calculate a score, which is 1 for control. And what I'm showing you here is a response to a number of different chemical samples that we've tested so far. And you can see that for specific samples, we're seeing nearly complete abolishment of the response to this painful stimulus. And this is ongoing research, but I can tell you that we've already got compounds that abolish the response behaviorally, which act on neural populations that we know to be associated with pain, and which also bind to receptors that are strongly associated with pain as well. So it's ongoing research, but you know, as I say, watch this space. And then one of the other areas that we've been working on is in collaboration with some of the other labs here at Whitney, the Ryan lab and the Martindale lab. And that's been trying to use some of these same techniques and some of these same ideas to understand how the nervous system works in other organisms, and in particular in these critters, which are comb jellies. Comb jellies occupy a really interesting position in the tree of life, which is that they diverged very early on from the rest of animals. So while the rest of animals kind of went in this direction, comb jellies went over here. And so they're in a really unique position to provide insights into how the nervous system of animals evolved, and how different nervous systems are put together and how different nervous systems function. And so we've been using similar techniques, using calcium imaging and population recordings to understand the sort of functional organization in ctenophores. And in the last part of the talk, I want to real quickly talk about some of the work that we've been doing in the direction of tools and methods environment, tools and methods development. A lot of our work is, or all of our work is motivated by biological questions, understanding how the nervous system is put together and how the animals respond to different stimuli. But almost all of it is enabled by tools and techniques. And so one of the things that we've been active in is trying to develop these tools and techniques and then make them available to the broader scientific community. And so real briefly, the software that we use for the behavioral analysis is developed in-house and released open source. The software that we use for the analyzing the neural recordings is developed in-house and released open source. And the multiphoton microscope is a sort of custom-built platform that we put together, and engineering specifications for that are also available to the scientific community. And you can actually just go to our website and download it. And in particular, we've been pushing recently on trying to develop even more advanced microscopy methods for asking different types of questions. And so one of the the sort of traditional limitations to volumetric imaging is the rate at which you can scan out volumes. Typically, volumes are scanned out by moving the objective up and down so that you can scan out different planes within the brain of the animal. This produces very high resolution, but it tends to be relatively slow. One of the alternative approaches that's been proposed is using a tunable lens. So in this construct, you put a polymer lens in the body of the microscope, and then electrical currents cause deformation of that lens, which shift the focal plane up and down, allowing you to record from different planes within the sample without moving the objective. Because you're not moving the objective, you can image volumes very, very quickly, potentially order of magnitude faster than would otherwise be possible. Now, this allows you to record not just calcium signals, but potentially voltage signals. So you can follow the processes or pathways within the brain with very, very high temporal resolution. Now, one of the challenges so far with this approach is that when you try and do this, it introduces blurriness into the image as you change the focus. Optical systems are some of the most precisely manufactured objects that humans produce, and if you introduce a deformable lens in there, it sort of messes up all of that precision. So you get blurry images as you change the focal position. You get different colors kind of blur, and then you also get blurriness at the edge of the field of view. And so what we did recently last year was we developed a theoretical framework to predict exactly how that blurriness would manifest. And we developed a sort of theoretical framework for compensating for that. And what we found is that if you use the designs that we suggest, that you can get an almost order of magnitude decrease in the aberration. This is the sort of optical error that you would see. So this is the sort of current state of the art. And if you use the design rules that we came up with, you get about an order of magnitude a decrease in the error. And that if you do a sort of full realizable implementation of that, you can completely eliminate the error. You can completely eliminate the aberrations, the blurriness that comes from changing the focal position, completely eliminate the color error, and completely eliminate the field of view error. And then the sort of future direction for this is we're currently looking at trying to apply these same ideas and principles to another microscopy technique, which is called the light sheet microscopy. In light sheet microscopy, the excitation doesn't come from the single objective, but instead you have a second objective that produces a light sheet to eliminate a single image plane. This has a lot of advantages, similar to multiphoton microscopy. It also resolves that problem with axial resolution. It's potentially much faster than multiphoton microscopy, but with all things, it comes with its own specific challenges. It's often quite difficult to prepare samples for it, and there are tissue light scattering issues. So a lot of opportunities, some challenges, and this is a sort of active research area for us to figure out how we can integrate some of the other technologies that we've developed with this microscopy approach, and whether or not we can resolve some of these outstanding issues. And a lot of these efforts have been fueled and supported by institution-wide effort at the Whitney Labs, which is the establishment of the Center for Biological Imaging and Discovery. This is really an institution-level program intended to establish a shared resource with cutting-edge advanced imaging systems to not just look at neuroscience research, but also developmental biology, regeneration, and aquaculture. And to not just offer these to researchers at the Whitney, but also to for this to operate as a community resource, for it to promote education and training of students in these methods so that they can facilitate broader adoption. All right, so that's to wrap up. I think that it's an exciting time to be doing neuroscience, because for the first time in the history of the field, it's possible to do experiments where you can record from not just one neuron or not just two or not just three, but from very large populations. And there's the potential to look at these very rich data sources using computational tools such as artificial intelligence and machine learning to really understand how some of the more complex circuits of the nervous system of the brain are put together. And so my lab has been looking at that within the specific context of the, And so my lab has been looking at that in the specific context of stress and stress-related behaviors. But there's really a phenomenal opportunity to move from a broad understanding of the nervous system and how nervous systems work to really a cellular level, fine-grained understanding for how these neural circuits are put together, how they're sort of computationally structured, and to understand how the emergent properties of those networks ultimately give rise to the complex behaviors of animals. And so with that, I'll wrap up, and I'm happy to take questions.